Okay, could somebody confirm you can hear me?
I can hear you.
Great. It's not working. My computer is going to be very slow because it's making the program for the lecture. I'm having a hard time getting the share to even work. There it goes. Let's close down a few things. Don't need that anymore. Hopefully, won't need that anymore. I suppose if I accidentally log out, I'll be in trouble. But um, let's go to the lab one. Remember that lab 00 and lab 01 must be turned in by Friday, 8 a.m., or else you'll be dropped from the class. And that's because I have to turn in who's attending and who's not by this Friday. Friday is also the last day to drop the class and then get 100% refund. Maybe a student can add. I saw that there's a date in there to get a 50% refund if you withdraw or drop from the class. I am not aware of this. I didn't hear anything about this, so maybe a student could comment on that. Anyone know anything about that? All right, I guess nobody knows about that either. Like I said,
If
I
you
go
if you
on.
go onto the Clark website and type in deadlines or important dates, it'll have a breakdown of the days that you can withdraw and how much money you'll get back for it.
I'm just surprised. I never heard that Clark had a 50% deadline, meaning you get 50% of your money back. I had never heard that, but I noticed it on there when I looked at the calendar this term. I'm just stating that it looks like if you drop by a certain date or maybe withdraw by a certain date, you can get 50% of your money back from the course. And I don't know anything about that. I'm just stating that. And to my knowledge, you only got 100% down back if you drop by this Friday. But apparently, that's not the case. And Clark College has a new system where if you drop later, you'll get 50% back. And I don't know anything about it. So all I'm stating is I saw it on the calendar. Okay,
It's the 27th, just to let you guys know.
fine. But like I stated, when did they start this? And do you know?
I don't know when they started it. I know the last three terms they've had it on there.
Okay, that's good. See, I didn't even know that, so I just discovered it this term. All right, let's move on to today's lab. We're going to be talking about lab one. Let me get the syllabus here. So we're going to be discussing lab one, the ubiquity of microorganisms. Oops. Let me remind you once again, you need to have lab 00 and lab 01 turned in by 8 a.m. on Friday, or you will get dropped from the class. Uh, the online plagiarism quiz is due 11:59 p.m. this Saturday. And by the way, on a quiz, you need to get it finished before the deadline because if the deadline hits, it'll kick you out of the quiz. And then all questions unanswered would be marked wrong. You must achieve 100% on the plagiarism quiz, or you will get a 0 on the 100-point infectious disease project. You should also read the infectious disease research project file in Canvas. It's the instructions. For next week, I'll be talking about that project next week. And so you should read about it. Let me see if I can show you where it is. There it is there. Infectious disease research project. You should read this. All right, any questions about anything? I can close that down. I don't think I'll need that. So let's go on. Lab Module 1, ubiquity of microorganisms. Microorganisms are found almost everywhere, and that's why we say the ubiquity of microorganisms. As all labs, there will be a little bit of reading, and this one's very little, maybe 3 pages on culture media, and then a page on obtaining a pure culture. There's a video clips, supplemental material in this lab. The learning objectives. Know that microorganisms are almost everywhere. That's number 1. 2, describe the purpose of culture media. 3, describe colony abundance and colony morphology. I thought there was another term, but I'm certainly drawing a blank on what it is at the moment. Define the following terms. What's inoculation? Ubiquity of microorganisms. What's something that's sterile? Isolation, incubation, in vitro, culture media, in which case there are 2 types. Oh, good. My computer will now work better. There's a broth media, and then there's a solid media. The broth media is a liquid media. We usually put agar into a liquid media to make it solid, although you can put other things like gelatin or starch in to make it solid as well. You grow the solid media on a plate or on a not a petri dish, a tube, test tube, to get either a slant or a deep. Let me show you. Well, I'll show you later. What's fastidious? A fastidious organism. What's a non-selective media? What does ubiquitous mean? And what's a colony? Microorganisms are organisms that are too small to be seen with the naked eye. Viruses and prions are included in this group, even though they aren't alive and they are not organisms. We will also include helminths, worms, in our studies, even though the adults are not microscopic. However, their infective stages are microscopic. The phrase ubiquity of microorganisms refers to the concept that microorganisms are essentially everywhere. A sterile environment is one free of living cells. In this lab, we'll investigate the abundance and diversity of microorganisms in common environments. We'll also learn basic lab protocols. There are different culture media, also called growth media. It is a sterile laboratory preparation that contains a variety of nutrients that are required for growing microorganisms in a lab, meaning in vitro. A culture media may be a liquid. We call those broths. They're usually in a test tube because they just spill too easily out of a petri dish. We can also add agar to the liquid media to make a solid media. We have a little video for you to watch on preparing pour plates. Note how the culture media needs to be sterilized prior to its use. And then there's an important question to consider. Why must a culture media be sterile prior to being used? We usually grow a solid media in a plate or in a test tube. We do not usually put a liquid media in a petri dish just because it'd be too easy to spill. You can sometimes do it, but most times in the microbiology lab, we do not. For a liquid media, we have it in a test tube shown here. And then for a solid media, you can have it on a plate, petri dish, or you can have it in a slant shown here, or you can have it in a deep shown here. And the difference is the agar is just across straight in the deep, and in the slant, the agar has a slant to it. The only media we're going to talk about today is a general purpose media. It is a non-selective media, usually complex in composition, and supports the growth of most non-fastidious organisms. A fastidious organism is one that does not grow easily, and you have to add certain ingredients to the media to get it to grow. Some general purpose media include nutrient agar with yeast extract, abbreviated NA, tryptic soy agar, abbreviated TSA, and nutrient agar, abbreviated NA. These grow most of the microbes that we grow in the microbiology lab. There are other types of media, but we will discuss them in another lesson. Whenever you're growing microbes in the lab, you should be appropriately labeling the microbes. And that is vital that the Petri dish or the test tube is correctly labeled prior to use. The following 5 items must be on the label: your name or your initials, the date, the type of media, you can abbreviate NA for the medium, the inoculum, that's what you're growing on the culture medium, and then the incubation temperature is optional. So here, there's the name of the instructor growing it. This is what they're growing, E. coli. This is the date, and then that's the media that it's growing on, TSA. For a test tube, you should write on a piece of tape and then stick the tape to the test tube. On a Petri dish, you normally write on the bottom of the Petri dish and on the edge of the bottom. You don't want to write here because that'll make it more difficult to see the microbes in the plate. You might be, I don't know, hiding the microbes or making it difficult to see. And we write around the edge so that you're not obscuring the most of the plate. Any question? Oh, you write on the bottom of the plate because if the lid gets separated from the bottom, the microbes will be on the bottom. So you want the information on the bottom. And this is important because I once worked in a lab where a lady was carrying a whole tray full of Petri dishes, and she had written all of her information on the lids. And then she tripped, spilling the whole tray, hundreds of dishes on the floor. And that whole experiment had to be repeated because no one knew what was in the dish and what the results were for that particular dish, because the lids got separated from the cells. Any question about any of that? All right. Inoculation is the placement or transfer of microbial specimens into or onto a sterile culture medium. So when we give you a culture medium in the lab, it will be sterilized before use, and usually it'll be sterilized in the autoclave. Incubation is the placement of the inoculated culture media in an appropriate physical environment for a prescribed period of time to allow the growth of microbes. You can have fancy incubators or simple incubators. A fancy incubator will include levels of oxygen and carbon dioxide, as well as temperature, humidity, and then obviously the time you're growing it for. Here we're looking at a fancy incubator where you put your cells to grow. Most of the incubators, actually all of the incubators in the microbiology lab are not this fancy. All they control is the temperature, where you put this in, set it out, and the humidity. What you do is you put water in this dish down at the bottom, and that makes the environment more humid. And that's important because that will keep the Petri dishes from drying out rapidly. They will eventually still dry out, but they can go in an humidified incubator for at least a week, sometimes longer. And if you don't have the humidity in there, they will only go for a few days. Our simple incubators only control temperature, humidity, and then the time that you grow it in. During incubation of a plate, the plates are incubated in the incubator upside down. Meaning this would be where we have our cells. This would be the plate. You put it in the incubator upside down. The reason being is the agar will be on the bottom and moisture will come out of the agar and it'll condense on the lid and then it'll form a droplet which will drop back onto the plate where your cells will be. And that will disperse the cells. It may not contaminate the cells. It could, like if the drop were to come down the edge here and then drop into the plate, that could contaminate the plate. But the drop should be sterile, but you don't want to spread around your different colonies. So we put them in the incubator upside down. Now the agar is at the top. And so the drop, if it were to form, would be on the top and drop down to the lid, which is not a problem because we don't have any cells growing on the lid. But also the agar will wick the moisture around, so a drop will never form and it will never drop down. And that's why we put the plates in the incubator upside down. Any question about any of that? Let me just state that the heat of the incubator causes the moisture to come out of the agar and condense on the lid. After growing your organisms in the incubator, you then bring out the plate and you look for visible bumps on the plates. These are colonies. Bacterial colonies have a rather smooth textured surface and a distinct edge, and a fungal colony does not. So there is a bacterial colony, distinct edge, smooth edge. Let me blow this up a little. Yep, I can't do it there. Let me do it this way. Bacterial colony, bacterial colony, bacterial colony. This is a fungal colony. You'll note that the edge is not distinct, and the edge is also hairy or, I don't know, fungal-like, fuzzy. Shrink that down again. So by looking, you can usually determine what's a fungal colony and what's a bacterial colony. Each colony is made up of tens to hundreds of millions of cells. Each colony has to have at least 1 million cells or it'll be too small to see. All the descendants of a colony may be derived from one cell. Obviously, a colony can be derived from 2 cells or a clump of cell, but some of them will be derived from one cell. And they will simply split from 1 or 2 or a clump of cells by binary fission until you get at least 1 million cells to form a colony. Here we have a little video showing you about binary fission. It's very short, so maybe I should go ahead and take a look at that.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
In theory, one individual cell can lead to the formation of one colony, yet a colony may be formed by two or more cells, being a clump of cells, in which case you cannot say that one colony here will be a pure culture. It looks like it's a pure culture, but you do not know that. And actually, about 30% of these colonies will not be a pure culture. Colonies are not visible in a broth culture because in a liquid culture, all the cells will simply grow together. There are often obvious visible colony morphological variations between different species of bacteria. For example, here you can clearly see that this is a different species than that one, and so is that one. So these are three different species, and that one's another. The different colony characteristics are shown in this chart here. You should know the simple colony characteristics. So the shape of the colony, you should know what a circular colony is, a rhizoid colony, a regular colony, maybe a spindle colony. I would not expect you to know the difference between a rhizoid and a filamentous colony, or maybe a filamentous and a regular colony, but you should know those. There, the margin is the edge of the colony. So a circular colony will have an entire edge, but a irregular colony will have undate or lobate or curled edge. And I would not expect you to know the difference between an undulate, lobate, and a curled margin, nor would I expect you to know the difference between a rhizoid and a filamentous colony. But I would expect you to be able to tell me the difference. Is it a rhizoid or filamentous, or undulate, lobate, or curled? You're getting the difference. So I expect you to be able to separate these two from these three, and then these groups from the entire edge. The elevation cannot be seen unless you take off the lid of the plate and look at the plate from the side. Otherwise, it's very difficult to get the elevation. You can have a flat colony, a raised colony, and that's like a flat mountain. You can have a convex colony, a pulvinate colony. I will not ask you to separate a convex colony from a pulvinate. And then an umbunate colony. Any question about any of that? Size, there's a punctiform colony, small, moderate, and large colony. A punctiform colony is a colony smaller than the point to a pen or a pencil. I was supposed to get a pencil here. I don't have a pencil. There we go. It's usually just a pinprick. Small is the size of the pen or a dull pencil. Moderate is between the size of the pen or the dull pencil and the size of the eraser. Large is a size greater than the eraser. Any question about any of that? Colonies can be smooth or rough, and believe it or not, this is what the students have the most difficult time telling me what's a smooth colony and what's a rough colony. The appearance, the colony can be glistening, that's a shiny colony, or a dull. Let's come back here. That's clearly a dull colony, and that's a shiny or glistening colony. Any question about that? That one is a rough colony, I'm not seeing other rough colonies here. So I would say that's smooth, that's smooth. I'm not sure about that one, but that's clearly rough. And these are all smooth. Pigmented means that the colony has color. To be non-pigmented, you have to be white, off-white, tan, beige, something like that. So these are all non-pigmented colonies. And then if it has color to it, yellowish. Actually, there's only two, well, yeah, there's only two clearly colored ones, the light yellow and then the dark yellow here. So that's a pigmented colony. So if you see color, it's pigmented. No color, it's non-pigmented. And then the optical property is the colony opaque, translucent, or transparent. Most colonies are opaque, some are translucent, only very few are transparent. These colonies are hard to see and they look like a drop of clear water. I don't have any transparent colonies. I don't see any translucent colonies there. Here we go. There is an opaque colony. And there's an opaque colony. There's an opaque colony. Here, this colony is translucent, at least on this part of it. Any questions about any of that? That would be an irregular shape colony, and that would be a circular colony. So is that.
I have a question. So when it's pointing to B, is that one colony grown on top of another one? Because you have irregular shape, then you have the circular shape inside.
No, that's one colony. I'm not sure what that is inside. Now, if you're talking about that one, I'm not sure if that's multiple colonies or one colony. That might be a colony right there growing on top of that colony. And it looks like these could be separate colonies here too. But that one there, that's the same color and actually the same type of edge as this colony. So I'm going to guess that that's one colony there. Okay.
Thank you.
Actually, colony B, what's it say? Medium, green, irregular shape, rough, flat. Yeah, that's implying it's one colony. All right, I think that's it for the colony characteristics. Two terms you need to know. Abundance. This is simply the total number of colonies on an agar plate. Diversity is the number of different kinds of colonies on a agar plate. So right there, let's go to an easy one. I'm not going to count all the colonies here, but if we look at right here, it's going to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. I don't remember where I put the line, so we'll go on with that. So something like 13 colonies in this region of the plate. That is the abundance of colonies in this region of the plate. The total number will be, that's 13. The total number will be something like 30 to 40 abundance of colonies on the plate. Now in this region here, I see clearly only two different colonies. So the diversity would be clearly 2. And that's assuming all of these are the same colony and that that one is also the same. I'm not positive about that one because that's a little bit lighter in color, but it might be just because it's smaller. But assuming that's the same as that, that's 1. And then there's the second. So there's the diversity right here is 2. Okay. And at most 3, if that's different. I'm not sure that's different from those. Any questions about diversity and abundance? You need to know those terms. I'll give an example here. In this picture here, what is the abundance of wild cats? Anyone? All you have to do is count the cats.
7.
7 is correct. 1, 2, 3, 4, 5, 6, 7. And what's the diversity of cat species?
2.
2.
2. Lions here and then cheetahs there. All right, so know the terms of this lab. You should know that we'll be doing, there's an interesting picture of somebody's hand, showing the microbes on somebody's hand. Probably Roberto, but I don't know. Let's go to the worksheet. Oh, that's not correct here. That has to be 8 a.m. Hopefully I got it corrected on yours. Oops.
On ours, it says the same before you corrected it, just so you know.
Yeah, that's not correct. It has to be turned in Friday at 8 a.m. It used to be lab module 1 used to be turned in on Saturday, but the registration office and Clark College got upset at us when we didn't allow students to join the class after we had the first lab on laboratory technique and safety technique. And so we've changed it. And now you can join the lab after the first date, as long as you join it by Thursday. But if you miss both Tuesday and Thursday, we can drop you from the class. And now lab 1 also has to be turned in by 8 a.m. to make sure that you are completing both labs. Okay? I'll try and change that on your page as well. Anyways, on lab 1, the protocol is we're not going to be doing any of this lab in real life. It's all going to be done for you in the lab, and it's going to be submitted online. In protocol lab module 1, procedure 1, we have procedure 1, what is in the air you breathe. Below is a nutrient agar plate that are exposed to air and then incubated for 24 to 48 hours at 30 degrees Celsius. And give me what the colony abundance on this plate is. Remember, just count the colonies, and that would be a separate colony. That's a separate colony. Right there, it's a little bit hard to figure out. Just give me a good guess. You don't have to be exact as long as you're close. And then give me the colony diversity on this plate. Once again, you don't have to be exact, but you must be close. Okay? And then answer step C, 1C. What step would you take to reduce airborne contamination of a sterile agar plate while you are inoculating it? So don't give me the steps before you inoculate it or after you inoculate it. What steps can you do to reduce airborne contamination while you have the plate open and are inoculating it? Procedure 2. What gets on your arms? Would you put your arms on the lab bench? Oops. We're going to swab the lab before you disinfect the lab bench and then after you disinfect the lab bench. And the first question is, did table disinfection work? That's simply a yes or no. And then B, what is the purpose of allowing the table disinfectant to air dry? You do have to think a little bit about that. Or why do we air dry the disinfectant? And there is a reason for that. You can go online and get an answer. I think it's discussed someplace in the lab module. Not clearly, but I think it is discussed someplace, at least hinted at. Procedure 3, what is on your hands? What we're going to do is look at the bacteria and microbes on your hands before you wash your hands, and then you touch the plate with your fingers, left hand or right hand. Then you wash your hands and then touch the plate. Now, obviously, you have to have the moisture on your hands similar to before you wash your hands and after you wash your hands. Otherwise, the moisture will wick the bacteria down onto the agar plate. And some students washed their hands and then didn't dry them, and they actually had more bacteria on their hands after they washed their hands than before they washed their hands. And so you need to have the moisture similar. And then answer the questions. Does hand washing work? And then 2, suggest one reason why hand washing did not result in the complete removal of microbes from your hands. It has something to do with the normal microbiota of your skin. If you obtain an answer following research, please provide a brief citation. You will need to think about this. Okay? Meaning it isn't obvious immediately, but if you think about it, you'll come up with the answer. Most students get this wrong the first time they submit an answer, but the second time, most students do get it right on the second grading of the lab. Procedure 4, when you kiss your significant other, what exactly are you giving him or her? What's on your lips and on your tongues? Here we have a probably an instructor touch their lips to the plate, and there you can see the microbes growing on the lips, and there the instructor touched the tongue to the plate, and there you can see the microbes growing on the plate. You should note this note I have above the picture, okay? And then answer the question, which plate has higher colony abundance, the lips or the tongue? And then B, why do you think this is the case, even though you might expect the abundance of both body sites to be the same, considering they're next to each other, and you probably often lick your lips? What might cause the observed difference in the colony abundance between the lips and the tongue? And I will give you a hint. You need to have 2 reasons for a difference. You can easily come up with 3, and I've had students actually come up with 5 possible reasons giving a difference. But there are 3 common, and I require you to come up with 2 reasons why there are a difference. And then C, why was the tongue plate incubated at a higher temperature than the lip plate? And it says in the procedure here, the lip plate was incubated at 30 degrees. The tongue plate was incubated at 37 degrees. Why are we incubating them at a different temperature? Procedure 5, are microorganisms in the soil? What we're going to do is take a little bit of soil, dilute it into water, and then put a little bit of the water on the agar plate, and then take a look at the growth growing on the agar plate. And what is the colony abundance of this slide picture here? Is it even possible to accurately answer this question? Please explain your answer. And answer the question, is there greater abundance of colonies from the soil compared to the lip plates? Okay, And note that the soil plate does have some tiny colonies, like right there. And the lip plate has mostly tiny colonies, but it does have a few large colonies too. All right, so answer the questions. I will be here to help you until the last student logs out. In the last lab, all the students logged out within 10 minutes, which is fine. You don't have to stick around to work on the lab. You can work on the lab anytime you want. But if you have questions, I will be here until the last student logs out to help you answer questions. Any questions? All right, if not, get to the lab. I'm going to stop the share and stop the video.